We've talked about a lot of kinds of membrane transport, so I want to summarize them all in a quick video here and have you do some work to review as well. What I want you to do first is think about what molecules we've been talking about and will they pass through the phospholipid bilayer, and why or why not. So pause the video to do this. So hopefully you got something like this. These highly charged molecules and glucose, which is fairly large and also hydrophilic, can't pass through very well. They have low permeability. There's some medium things here. Steroids will come back to. They can go through fairly easily. Water, these are enough to make a difference, right? Like water can go through via simple diffusion. However, aquaporins are also helpful. Really small hydrophobic molecules can pass through very easily. Large proteins cannot pass through the phospholipid bilayer. So things that can't pass through this bilayer need either a protein or vesicular transport, some other mechanism. And this can then alter the permeability of the membrane. So the next what I want to do is review the types of cellular transport in a flow chart. So these are the two main types of cellular transport, and we've been focusing on this one, going through or into cells. I mentioned in one of the videos that things can pass in between cells as well. This is called paracellular transport. That wasn't the focus of this, but it's good to know that some things do go that way. It does depend on how closely spaced the cells are. So we've been focused on this way over here. So either through the cells, so that's transcellular, or into the cells, right, to do something. So the different processes we've talked about are passive, first of all. These are things that don't use ATP. We're going down a concentration gradient, which means there's a drive, a natural drive for these to happen. The three types of passive transport are simple diffusion, right through the phospholipid bilayer, if it's something that can go through the bilayer. Facilitated diffusion, for large or charged things that need a protein to go down their gradient. So the difference between these two, still a gradient, but can the thing go through without a protein or not? What matters for transport across a membrane is both the gradient and the properties of that chemical, whether it can go through or it has a protein it can go through. Osmosis is a special type of diffusion because it's the diffusion of water, and it uses both simple diffusion and facilitated diffusion because water can pass both through the membrane directly and also uses aquaporin. But it's a separate category because it's good to study it on its own, and things are just a little different with it, right, in terms of water moving from high water concentration to low water concentration, or from low solute concentration to high solute concentration, and it's water movement. So the other big theme of transport is active transport. So this would be up a concentration gradient. There's not spontaneous drive for these. So we need to have some sort of energy input. And the two categories are active transport, ATP pump, and vesicular transport. Vesicular uses ATP. I didn't talk about where, just know that it does require energy. Active transport is ATP pumps. Sodium potassium pump is one type of that. Notice I don't have secondary active transport on here just because that gets a little messier. That's using the gradient from this pump to do other work. So vesicular transport, then there's exocytosis out of the cell and endocytosis into the cell. And I am going to add for myself here one more. What am I missing? This is a learning check. Pinocytosis, as well as receptor mediated. And then for exocytosis, something going out of the cell, same idea either way. Secretion, though, is something that's going to go into somewhere else in the body, probably. Or excretion is usually waste that would be going outside the body or into a digestive tract. Okay, one more. So for this, what I want you to do is fill this table out. What is the energy source, if any, for each of these? Is a protein required? And what molecules are transported via this mechanism? So pause to do this. Okay, so if you didn't put kinetic, that's fine. Kinetic energy means it's just within the system, right? There's no external energy required. If you had put spontaneous for that, that would be fine. Spontaneous, spontaneous, spontaneous. Then active transport and secondary both require energy. Secondary, it is a ion gradient that's actually providing that, maintained by ATP. So it's kind of like a company secondarily uses ATP. Table like this is not meant, right, this gets a little tricky about where this falls in. The idea is to help you understand it, and the idea behind secondary active transport is it's complicated. So describing it like this is great. Is a protein required? No, yes, sometimes, aquaporin or not. Yes and yes, so these two, yep. And then what are examples of molecules that move via this mechanism? This has to be our small hydrophobic. This is going to be those things that can't pass through the membrane on their own, but still typically have concentration gradients. Not always, right? Glucose doesn't always have a concentration gradient. Sodium doesn't always have a concentration gradient, but it can. This would be carrier molecules. This would be channels. Water, ions typically for active transport, secondary active transport, glucose, another example is amino acids as well. So things that sometimes would have to go against their concentration gradient and require a protein. Okay, so that's cell transport. The one last video for this week is on cell signaling. So a different topic, but still related in terms of membrane proteins.